A research group from Gifu University developed innovative therapeutic drug candidates that can penetrate the cell to stop proliferation and metastasis. Dr. Ryo Honda, Associate Professor from Gifu University, will provide a detailed explanation of the research.
Approximately 30% of cancers harbor an activating mutation in the RAS protein. Accordingly, the development of novel RAS inhibitors has been the focus of many studies worldwide. We successfully developed a new RAS inhibitor using a novel strategy based on the protein chemistry in which I could exercise my expertise.
Drugs that target a particular protein or macromolecule are commonly low molecular weight compounds that inactive the target protein by fitting perfectly into small cavities on the protein surface. However, RAS proteins have no such features on their surface. Therefore, Dr. Honda considered developing a method with the aim of creating a high molecular weight protein harboring a RAS binding domain RBD that is larger than the RAS protein itself, thus allowing for specific binding to a large area on the surface of RAS protein. To achieve this, the RBD needs to get inside the cell.
As I proceed with my research, I came up with the idea of synthesizing a number of cell-penetrating proteins. A previous study had reported that when peptides capable of penetrating the cell membrane are fused to a protein known as ubiquitin, each of the proteins penetrates the cell membrane. I thought that since RBD has a structure similar to that of ubiquitin, it could be penetrated into the cell by fusing it with a peptide in a similar way.
To achieve this, one needs to carry out difficult experiments involving synthesis and purification of a number of fusion proteins before administering them to cells. 11 types of RBD and 12 types of cell-penetrating peptides were selected based on a review of previous studies. Fusion proteins combining the various RBD and peptides as mentioned above were synthesized and applied to cancer cells. They were then evaluated 51 times after 6 months of trial and error. Dr. Honda has been successful in developing RAS inhibitors that suppress RAS proteins.
That is pan-CRAF-V1.
By comparing pan-CRAF-V1 with inhibitors reported in previous studies, it was revealed that the new inhibitor exerted inhibitory effects on multiple mutant RAS proteins, demonstrating its superiority over other previously characterized inhibitors.
We are currently proceeding with evaluation in mice. In experiments conducted on cancer cells in vitro, the RAS inhibitor that I developed inhibits several mutant RAS proteins, but its effect in mice has not yet been confirmed. In addition, more research needs to be carried out to find out whether the new RAS inhibitor is degraded in the body or excreted in urine. The underlying mechanism needs to be determined, and further improvements may be needed for it to inhibit RAS in vivo. RAS inhibitors are considered difficult to develop, and we evaluated them as a therapeutic agent so that they can be used not only on cells but also in organisms such as mice and eventually in clinical settings. Because of our pioneering efforts, we also received Nobel support, and the research environment has been enhanced for newcomers from other fields of specialty. I want to carry out path-breaking work in which I am confident of success.